So good afternoon, everyone. I'm Ram. I work at Barts in London, part of Team G. Today, I think I'm the last science talk of the day, and I'll be talking about the genetics of MS. So starting off, does MS have a genetic component? And one way to look to see if genes are involved in a disease, you look to see if the disease clusters in families, so more than one family member has the disease. And people have been looking at this for over 100 years. For example, this family described in the 1930s. So this is the family tree. The shaded individuals are people with MS. And this family has 8 affected individuals over 3 generations. And so it suggested that maybe there's a gene for MS in this family. It comes from the grandfather and it's passed down to subsequent generations. And that leads to MS. So there's a genetic component to MS. But this is only one family, so it could be a chance finding. And it could also be whatever this family does together, so what we call the familial environment. Families tend to eat the same things, they go on holiday together, they get sick together. So whatever this family isn't doing in particular could be increasing their risk, rather than it being a genetic effect. Now, in order to remove the element of chance, researchers have looked at many families. And so this is a study from Canada looking at over 30,000 MS patient families. And what it clearly showed was that the more related you are to an MS patient, the higher your risk of getting the disease. So if you're someone in the general population who doesn't have an affected relative, your risk of getting MS is about 0.1%, so 1 in 1,000. If you're an aunt, uncle, niece, nephew, or cousin, so a secondary relative, your risk rises up a little bit, so it's between 1 and 2%. If you're a first-degree relative, a child, parent, or sibling, your risk of getting MS is about 3 or 4%. And risk rises all the way up to about 30% if you're genetically identical or monozygotic twin of an MS patient. So this kind of implies genetics. The more related you are to an MS patient, the higher your risk of getting the disease. But it doesn't necessarily prove genetics. As you can imagine, if you're living with your sibling, you tend to probably more likely to do things more similar than as you would with your aunt or uncle. So it could still be the familial environment that's increasing your risk. Now, in order to separate out genes from the environment, people have looked at what's the risk of developing MS if you're living with someone who's got MS but not biologically related. So if you're an adopted family member, if you're a spouse, or you're a step-sibling. And what these studies have quite clearly shown is that if you're not biologically related to an MS patient and you live with someone with MS, you've got no increased risk. So whatever increases risk in families is almost entirely genetic. And so MS is what we call a complex disease. So this means there's no single risk factor. There are many risk factors. Some of these are genetic, and they only increase your risk. If you have a gene that's related to MS, it increases your risk. It doesn't mean that you're going to get MS. There's also environmental risk factors. This is anything that's non-genetic. And there's probably the element of chance. These things need to occur perhaps in a certain sequence for MS to develop. Now, if we know genes are involved, how do we go about finding them? So just to recap some biology, we all have 23 pairs of chromosomes, so 23 from a mother, 23 from a father. Chromosomes are made up of DNA. DNA codes for genes, and genes codes for proteins. And proteins are essentially what make us what we are. Watson and Crick, as Julian mentioned earlier, solve the structure of DNA. It's a twisted ladder or a double helix. And the rungs of that ladder is essentially the genetic code. So it's a series of letters A, C, T, or G. The rungs of that ladder can vary between people. And we call this single nucleotide polymorphisms, or SNPs. And in order to find disease-causing SNPs or disease-causing genes, we try to compare the frequency of these rungs between people with MS and people without MS. For example, in this example, some people can have a CG rung, whereas other people may have a TA rung. And looking at the DNA of people with MS, we look to see the frequency of the CG rungs as in people with MS and compare it to people without MS. And in this example, the CG rung appears to be more common in MS patients. So we'd say that CG rung is a risk factor, or that's a gene involved in MS. Now that all sounds quite straightforward, but there's a problem in that there's 30,000 genes and 10 million changes in the rungs that we know about so far. So it was thought to find genes associated to MS, it was going to be like finding a needle in a haystack. It was going to be pretty hard. But there was some early luck. So when these studies started in the late 60s and early 70s, some researchers in Scandinavia lucked out and found association with a gene on chromosome 6 called HLA-DRB1. This gene is involved in the immune system. It's a protein that's involved in recognizing your own proteins versus foreign proteins such as viruses or bacteria. And this fits in with what we knew about MS. So this was quite a nice link between pathology of MS and genetics. Now, after that initial success, researchers spent many decades trying to find additional genes for MS. So things happened, prime ministers changed, but like trying to find weapons of mass destruction in Iraq, we couldn't find any new genes for MS over the subsequent decades. But recently, there has been what's been known as a genetic revolution, and this has been almost entirely driven by changes in technology. So back in the 1670s, you'd look for changes or SNPs, changes in the DNA, a single SNP at a time. It would take weeks or months to look at that single SNP, and you'd do it manually. But with developments in technology, we can now look at 5 million SNPs at a single time, all through automated processes. And while improving technology has occurred, it's not quite CSI standard yet, so it does take a few weeks or a few months to analyze genetic data. And in terms of MS, so some great strides were made last year in a paper published looking at the DNA of 10,000 people with MS and 20,000 people without the disease. And the researchers identified about 60 new genes for MS. Some of the names are listed in there. They're not very exciting, but they all kind of point to, they're all involved in the immune system, and they all point to the immune system as the primary thing going wrong in MS. In terms of what these genes have on the effect on how much your risk is increased, the gene that was identified in the 70s, the HLA-DRB1 gene, that gene has the strongest effect on MS risk. So having 2 copies of the risk variant of that gene increases your risk by several 100%. And this is in comparison to the new genes that have been identified, who only increase your risk by 10 or 20%. And so now with all this data, so we now have 60 genes that we know are involved in MS. The question has been, if we could put all these genes together and use this as a test, can we predict who's going to develop MS? And prediction, we use these scores called sensitivity and specificity, and plotting this on a graph, you can use that as a test to see whether the test is good, medium, or really not useful at all. And so that's what the results look for, like in MS. We're bordering on it, using all the knowledge from all these genes being a medium risk, MS prediction tool. So it's not something we can actually use at the moment. These genes won't be able to tell us who's going to get MS. Now, with Team G, we're wondering if we can actually develop this tool, incorporate things other than genetics to see if we can actually develop a predictive tool to see who's developing MS. Ultimately, this is so we can identify people at high risk and then initiate prevention strategies to stop them from ever getting the disease. One of the things that we're looking at is something called epigenetics. So all our cells in our body have the same DNA, but they're doing different things. So a nerve cell is very different to a muscle cell, and a muscle cell is very different to a stomach cell, and a stomach cell is very different to a liver cell. And those differences almost entirely arise from difference in the epigenome. And the epigenome encompasses a number of different modifications, and that includes DNA methylation, which is a chemical change onto the DNA sequence. And I mean, epigenetics is a relatively new field, so it's only kind of exploded in the last 3 or 4 years. And it's primarily because of interesting studies, such as this one in using animal models. So these identical animal models have different coat colors, and there are different risks for cardiovascular disease. And this was the differences are almost entirely to their epigenome, to DNA methylation differences. And you can change, their risk could be changed by their environmental exposure. So changing their diet could change their epigenome and then change their coat color and change their risk. Now, we've been looking at genetically identical twins discordant for MS. So one twin has MS, the other twin doesn't. We've been looking at their DNA methylation patterns to see if there's any differences. And so what we've found so far, there are strong differences. So there's differences in methylation between twins who have MS as compared to their co-twin who doesn't. So this may also be an additional factor on top of genetics that can predict who's going to develop MS. And so just to end with, will we ever be able to predict MS? We believe we will be able to. It won't be a single thing that we're going to have to use. It won't be just genetics. We'll have to incorporate genetics with environmental risk factors such as vitamin D deficiency, whether you've smoked, whether you've been infected with the virus, Epstein-Barr virus, and also probably other things such as epigenetic differences and gene expression differences. That's my talk. Thanks for listening.